Good evening and welcome to this evening's Gallagher Colloquium lecture series presentation. Again, I'm not going to introduce myself. I am Rachel Lauer, your MC for tonight. And we've organized tonight's event to be actually a hybrid event to continue to engage with people remotely who choose to remain home and attend online from around the world or around Calgary, and also to welcome audience members to our campus to have this experience in person. So whether you're home and enjoying this online or here in person, we're glad you've decided to spend some of your evening with us. So thank you all for coming. I'd like to mention that tonight's Q&A session will include questions from both online participants as well as in-person audience members. We'll probably stagger between the two just to give everyone a chance to ask their questions. Before I introduce tonight's speaker, I would like to take this opportunity to acknowledge the land that we are privileged to live and work on today. These are the traditional territories of the people of the Treaty 7 region in southern Alberta, which includes the Blackfoot Confederacy comprising the Siksika, Piikani, and Kainai First Nations, as well as the Tsuut'ina First Nation and the Stoney Nakoda, including the Chiniki, Bearspaw, and Goodstoney First Nations. The city of Calgary is also home to Métis Nation of Alberta, Region 3. We also acknowledge we are standing on land that is situated immediately adjacent to the Bow River, a river that has shaped this land and its people for generations. The traditional Blackfoot name of this place is Mohkinstsis, which we now call the city of Calgary. The Department of Geoscience has had the privilege of organizing the Gallagher Colloquium lecture series for many years. These presentations continue to give our department and the Faculty of Science the opportunity to welcome and host world-class researchers who all share a passion for science, scientific research, and exploration. I'd like to take a moment to recognize the Gallagher family for making this series possible through their generosity and vision for deep connections between the university and our community. Their support through this series allows us to promote and build public awareness for science and scientific research. And it helps people understand why science is fundamental and important not only to our lives, but to the world we all live in. We're grateful to be able to host this important lecture series. Okay, I'm pleased to welcome this evening's Gallagher Colloquium presenter, Dr. Heather Graham. Dr. Heather Graham is an organic geochemist and research physical scientist in the astrochemistry laboratory at the NASA Goddard Space Flight Center. Her research focuses on the fundamental scientific development of tools and techniques that can be used for agnostic biosignature detection, both in situ and as part of sample return. Thank you. That's Heather. Here she is in person and in a cave. Yeah. With a diverse background, very diverse background in paleobiology, ecology, analytical chemistry, and statistical modeling, Dr. Graham combines expertise in the expression and preservation of chemical biosignatures with an understanding of the constraints of space flight instrumentation. Dr. Graham is a member of the OSIRIS-REx Soluble Organics Analysis team, a co-lead for the Network for Life Detection, and a fellow of the Canadian Institute for Advanced Research, also known as CIFAR. She received her PhD in geosciences and biogeochemistry from Pennsylvania State University and an AB in chemistry from Occidental College. Oh, there you go. Something that did not come up in a Google search of Dr. Graham is that she also co-wrote and co-directed a rock opera in Baltimore based on the life story of Katherine Johnson called Determination of Azimuth, which is the first 3 words in a paper that she wrote. Katherine Johnson, not Heather. And this came out before the movie on the subject you may have heard of called Hidden Figures. So please welcome me in joining Dr. Heather Graham, who will discuss time machines and spaceships, building search patterns for life detection.
Thank you, everyone. I'm looking at this and wondering if we can get rid of the thing at the top that says recording on.
Yeah.
Okay, cool. Because there's words up there. You might want them. Thank you everyone for coming out tonight, for braving the cold to come and hear this strangely tantalizing title of a talk that I'm hoping will engage all of you. It's been a real delight today to spend time amongst geologists and planetary scientists, because I really feel like that's my home discipline, even though you're going to hear a lot of stuff that isn't planetary science or geology. But I'm hoping that there's something in this talk that engages everyone. I know we have a really diverse group of people here. So in this journey that we're going to take, either in a time machine or a spaceship, we're going to be going out into the solar system. We're going to be, on this grand tour of all of the worlds that are part of our solar system and thinking about the possibility for life on those places. Because we here on Earth live in this vast sea of stars, and most of those stars have clusters of planets around them. And even though we can barely see those stars, we know, because we understand the physical laws that direct energy and matter, that we can kind of predict what we will see there, what the geology will be, the atmospheres, the kind of energy constraints that might be on that planet. And so we're going to look at that and see, knowing all that, can we understand what kind of life might be on those worlds? And I'm starting my talk with this here, which Carl Sagan called our family portrait. I don't know if all of you can see that tiny speck that I'm pointing at, but that's Earth, that's us. That's all the life that we know about is right there on that tiny speck. And that's unbelievable to think that that might be the only life out there in the sea of planets in our universe. That tiny speck picture was taken by the Voyager spacecraft as it was leaving our solar system. It was speeding past Neptune, and it turned back around to take one last picture of all of us here. And there it is. So I know probably a lot of people in this room don't think about life, or at least don't think about it as much as I do, but everybody probably has an idea of what they think it looks like. And so you probably, as a person who is alive and observing life, have ideas about how we can look for life, or at least what you think life is. And that makes sense, because we live on a planet that's absolutely saturated with life. Everywhere we go, we're finding it. In really improbable and annoying places, we can still find life. At the bottom of the seafloor, up in the arid deserts, there it is. In the ionosphere, not in the ionosphere, in the upper atmosphere, we're finding it on dust. And we can look at this and say, just from observing all of these life forms, we can see that there are traits and features that all of these life forms share. And so even these elephants are sharing a lot of the same traits and features in biochemistry and molecular tools as these microbes here. They're wildly different scales. They have very different lifestyles. They have very different ways of navigating the landscape, the habitat they live in, but they have all the same similar biochemical machinery. And that's because you and me and elephants and microbes all share the same common heritage. We're all related. We all come from a single long ancestry of life on this planet. But when we look out into the solar system and think about other planets, we can't assume we're going to have a common heritage with all of those other potential organisms on all of those other planets. If we think about the fact that it takes 7 years to send a spaceship to Titan, what's the likelihood that we share ancestry with any organisms that might be in that moon system? So when we think about life detection in the solar system, we have to get creative and start thinking beyond our common heritage. What would an organism that lives on a world like Titan even look like? This is a wildly alien landscape by comparison to what we, organisms on Earth, are optimized to survive. These are hydrocarbon lakes. This isn't water. This is ethane and methane. It's in a weird landscape. So if we think about our heritage and our ancestry and our machinery that we all share, this is not a place where we could survive, but we can start to think about the landscape that is there and imagine what might be able to live there. If something's going to live there, for example, it couldn't share the same solvent that you and I all share to run our reactions, and that's water. So we have to move beyond thinking about this common heritage and imagine, what is the possibility space for life? What is physically possible for life to actually exist in? And I know this is not very readable, and I'm really sorry, so I'll just read it for you. And what I'm saying here is, can we search for life without presupposing familiar forms and a similar molecular framework? We're going to work through some of the basic features of life and imagine how we can think about generic traits that we can understand when we look at other planets. And I think a really good place for us to start in this search is to think about the core discipline that has really led us in astrobiology to think about life, and that is paleobiology. And we take a lot of our tools and techniques and the way we organize our discipline in astrobiology from paleobiology, and many of us in astrobiology, like myself, come from this world. And this is a convenient way for us to start thinking about the search for life, because even in the distant past, life was so different than what we have now in our modern world that you have to take those leaps of imagination to understand what that organism was. If you look into our deep past, even on our own planet, it really encapsulates that old adage that the past is another country. If I were to plop you down on the beaches of Alberta, where this animal once lived, you probably wouldn't know if I had sent you there in a time machine or a spaceship, because it's so wildly different. Similarly, we can look at these remnants of antiquity, these ancient animals or ancient creatures that have been on the landscape for so long. This is stromatolites, for those of you who don't know. These things that look like rocks are actually colonies of microbes that are accreting dirt and sand from the water and building these structures. This is the lifestyle that has been on this planet for hundreds of millions of years, billions of years. And we can find these in the fossil record, and we can find these off the coast of Australia. We can even think of these, you know, if you're imaginative, as the first cities. That these are huge communities of organisms all living together. And you look at that and you can't even imagine the antiquity that that represents. And you have to disengage your conception of what life is to recognize this, even in the modern. I also want to caution everyone when thinking about paleobiology and life, to remind everyone that you can't look at the distant past and predict what something's going to look like in the future. If we go back to the Rhynie chert, which is the earliest fossil of land plants, and see these tiny little creatures, that's a 3 centimeter scale, you know, rootless plant-like things living on the surface of the earth, you wouldn't look at those and predict that someday in 400 million years, they're all going to look like this. Because evolution is a pastiche of all of the events that are happening with the organism in response to its community and the habitat that it finds itself in. And that's unpredictable on long time scales. When you look at an organism, you can't know what's going to happen to that organism over time. So this is a way of saying that when we look for life, we have to recognize it's a coordinated response to its environment. And this is a way that we can start to use this idea of life and its relationship to its environment to understand how we'll look for it, even if we don't know what its biochemistry is going to look like, because we're not using that analog of ourselves. And I like to tell people, especially when we're talking about, you know, folks in other disciplines who don't think about life all the time, that life can be recognized by first understanding its absence, by developing a knowledge of the expected abiotic expressions given the rules of a physical system. So my search for life, all of the work of people who are physical scientists or geologists in this room, I depend on the knowledge set that comes from understanding abiotic systems. Because when I look for life, I have to understand first what the constraints of that abiotic system are before I can tell if that thing might be life. I like to use this example of a definition of life within the context of that idea, that life is an energetic response to an environment. I like to describe life as thinking about Newton's cradle. And when a Newton's cradle is plopping along, it's following physical laws. But if you see this ball, this Newton's cradle, it's doing something slightly weird. It's rising up higher than it should. And if you think about that little bit of extra energy in that system, that's an indicator of life. That's the kind of metaphor that I use when I look at abiotic systems. I look for something that represents some energy input that into that system that might be life, because that's how we can recognize it as a chemical or physical effect. It's an input of energy in a system. So I'm also going to tell you a lot of really boring definitions of life, but that's only because we don't have a definition of life. And these are operational definitions that help us in this search for life. And a really basic way to think about life is that organisms are open systems with fluxes of matter and energy far from equilibrium thermodynamics. That's a thermodynamic way of saying the exact same thing I said with the Newton's cradle. And we can look at it this way as thinking about life, bringing stuff in, elements in, sequestering them, secreting other things, and doing this in a way that is not what would be expected if just thermodynamics was running in your system. We can also remember that organisms arrange matter and energy into highly ordered structures with intrinsic complexity. And we can look at a single organism and recognize this highly ordered structures at various scales. Here I'm showing a leaf where you can see this beautiful venation and all of this structure, which is something that would be very unlikely to arise without life there. We wouldn't expect a bunch of carbon to do that. And then at the small scale, there's an even more complex structures of chlorophyll inside little packages inside that leaf, doing the metabolic business of the cell. So how do we look for life when we don't have any of these convenient preconceptions from looking at life? And this is what we do in my lab is we think about how to decipher biosignatures from the abiotics context. I'm going to use the word biosignatures a lot. It was in my bio. And so I just want to give a brief definition of what that means when I say biosignature, because it's a little bit of jargon that you've probably heard before. A biosignature is just an indication of past or present life. It can be as simple as these fossil bones, or it can be as complex as TV's favorite biosignature, the DNA molecule. But both of these are highly derived states of matter that were wildly improbable about the energetic input of life. We can also think of a biosignature as an imprint on an environment, like the many biosignatures that we've left on the surface of the moon that say to anybody from around the solar system who visits our moon, that something alive was there. That's not a pattern that's going to arise without life putting an imprint on that environment. The most basic way that we could start to think about the business of life and metabolism is by using Albert Szent-György's definition that life is nothing but an electron looking for a place to rest. This is a way of really conceptualizing the energy that I'm talking about. And what we see here in this graph is a solution of minerals, and the sterile solution of minerals is shown in black, and the white diamonds are showing the voltage changes. That's electrons transporting outside of a cell in that solution of minerals. So just here, you can see those electrons that tell us there's life there. This is a way you can even look for life on the sea floor. This is an observatory that's been put on the seafloor where they're doing just that. They're looking for electrons. They're not sequencing. They're not taking pictures. They're not analyzing chemistry. They're just looking for electrons because they know that there are organisms there that are doing something wildly improbable in that abiotic context. They're transporting electrons outside themselves and reducing metals there. And so finding reduced metals in the abiotic marine context is wildly improbable. And so when you see that strange thing, you think, oh, there was energy there in that system that might have come from life to make that happen. But we don't need to steam around the solar system looking for electrons, because the redistribution of materials also represents energy in the system. So that's what it says up there. You can see. So evidence of disequilibrium and energy transfer is another way that we can think about looking for life. If you are a sedimentologist or a person who looks at rocks, you can see this rock here has this strange pattern. That strange pattern would be wildly unlikely if this was just a sediment resting at the bottom of a river without any life influencing how that rock was forming. But this geologic material has been influenced by the presence of biofilms in that stream bed, and that's that pattern that we see. And so geologic materials are accreting and stabilizing in ways that have distinctive structures and fabrics that tell us something alive was there that redistributed matter in such a way that we know there's energy in the system. The trilobite is another example of that. A body fossil is a way of saying matter was redistributed in this strange way that is life. And I would also like to point out that a lens cap is also a great biosignature. Those are highly unlikely to arrive on the landscape without life being there. We've been looking, we found no lens caps in the solar system. So redistribution of materials represents energy and a selection mechanism. It isn't just energy doing things for no good reason. You are all alive right now because molecules in your body are performing reactions that have been selected to be advantageous to keep you here. We can also use this really unromantic definition of what life is by saying life concentrates scarce elements into discrete metastable entities that are geochemically distinct. So it's, as I was telling Rachel, like, you're one of my favorite metastable entities. She's great. She's so geochemically distinct. And this is an intuitive thing that we all know and can understand, is that organisms are accumulating elements in the environment as part of their structure and excluding other things. We're just thinking about a single-celled organism here, but you are doing the same thing right now. You are excreting certain things as you breathe. You have other things that are accumulating in your organism structure. So this is one way to start thinking about life. And we can see this at the micro scale. And we can see how those geochemically distinct things don't look like everything around them. This is a cell that has been analyzed with nanosims. And so what we can do with that technique is this well-lit part right here is something that shows us carbon bonded to nitrogen. And in the cell, of course, this is an organic thing, we see lots of those bonds. We see lots of those carbon-nitrogen bonds, and we don't see a whole lot in the fluid around that cell. Well, we expect to see that, right? Because this is an organic critter. We're all made of organic molecules, reduced carbon molecules. So we expect that. And that doesn't sound a whole lot like I'm looking outside of the realm of chemical heritage to find life, right? Well, you can actually do this with other elements. You don't just have to use your basic elements. We can think about phosphorus. So in this cell, using that same kind of technique, we can see pockets of phosphorus that have been accumulated inside that cell that are very different from the abiotic context. You're not seeing a whole lot of free phosphorus in the water, but you are inside that cell. But we know that phosphorus is something we also associate with life. You can do the same thing even with something as weird as copper. Copper isn't something that we normally associate with life, but in the cell, we see an accumulation of copper that's still greater. You know, there is copper in this fluid, but still greater than what's in the fluid. This is probably part of an enzyme, some sort of cofactor, or maybe it's just part of the organism and there hasn't been a particularly good reason for it to excrete it. So all of these things are just showing us that life concentrates these scarce elements into these metastable entities that are geochemically distinct and can tell us there's something special happening here that's not happening out in the fluid. And we can recognize that as life, and if we know enough about what the abiotic context, the fluid of that organism would be. Another way, you don't have to just think about this on the single cell way, you can think about this on the surface of any planet. What I'm showing you right here is a little stone crop and a little plant. And these are accumulations of carbon on a silicate landscape that are anomalous. These represent huge accumulations of carbon relative to everything around them. Another way to think about this is as you're flying over the desert in a plane and you see all the sagebrush, those are anomalous accumulations of carbon in an otherwise silicate background. And you can say, wow, I think that's life. You and I are anomalous accumulations of carbon on this relative to the world we live on. And in the spirit of the last example I gave of the lens cap, even our phones are wildly anomalous accumulations of rare earth metals that we've dragged up from the depths of the earth and stuck into iPhones. And even if you can't find me, you find my phone, that's a biosignature. Life was there. This is something that you can even see at planetary scale. I'm sorry this is really fuzzy, but this is a 50-kilometer scale bar here. And what you're seeing in the top panel is the coast of New Jersey. And in the bottom panel is the coast of New Jersey experiencing an algal bloom. This is a huge flux of energy and matter that represents life. All of this carbon is accumulating as these animals go through growth bloom and are sucking all of that out of the water and creating this huge anomalous structure of all of this elements that make up life in a very different proportion to what we see in the fluid around it. And something that we've been working on in my group, and I've been working on with colleagues at Santa Fe Institute, is this idea of ecological stoichiometry, that these fluxes can be generalized across really big areas to try and recognize the presence of life by thinking about the relative abundance of certain elements in a thing versus the environment around it, in this case, a fluid. So we take this idea from the idea of the observation of the Redfield ratio. For those of you who don't know, this is a phenomenon in the ocean where just about anywhere you go in our ocean on Earth, you will find the same ratio of nitrogen to phosphorus in the waters all across the Earth. And this is the pattern that's being driven entirely by biology. Biology is sucking up that nitrogen and phosphorus so that this observation we have about this ratio is really the cells themselves. And we can see that reflected in the bulk chemistry of the seawater. And it's not just nitrogen and phosphorus. We're also seeing that same idea of accumulation of certain elements in organisms versus their abiotic context as they build their metastable entities in many other element systems. So here above this line are things that we're seeing that are being absorbed as part of an organism, and then things that are secreted, chlorine, sodium, magnesium. So now they are in greater abundance in the seawater. So this means that we can look at a particle in the seawater, look at how much of a certain element is in that particle versus its abiotic context, and start to try and understand if this represents something that's alive. Because even when we look at these particles, in this example, the blue particles are calcite that's made as part of a phytoplankton's shell. And the red is abiotic calcite that's just precipitating out of the water, perfectly naturally following just chemical laws or physical laws. And we're seeing different amounts of trace elements in each of those particle reservoirs because that organism was sequestering some of these things as part of its cell. And this is some modeling work that we've done where we started looking at the relationship between particle size. We don't care if it's a grain of dust. We don't care if it's an organism. Just if you look at particle size, can you use the stoichiometry, the relative abundance of elements in those particles as a way to understand life? We started from the idea of scaling laws. This idea that the relative amount of nitrogen and phosphorus inside a cell differs with the cell volume. And that's because small cells are more dominated by things like DNA that have a ton of phosphorus, and large cells are mostly a lot of ribosomes and other things like that that might have more of other certain elements. So we can see this, and what we really see is that in the most of the cases of the cells that we can imagine living in the ocean, they're not at the Redfield ratio. So this tells us something about what the population size of cell volume that's probably in our ocean is represented by. We can then model that, start to think about the environment's entropy ratio versus the cellular entropy ratio, and how we could just look at the particle sizes that we see in a fluid and understand, are they representing the same as the water, or are they falling off that line in a way that we expect, given the kind of reservoirs of macromolecules that would be in them. And the next step in all of this is really thinking about how particle size relates to life. Particles, if you just have a sediment trap out in the ocean and are collecting all of the particles that show up, you're going to get dust, you're going to get minerals that are breaking down, and you might get a few cells. But the sizes of those different reservoirs are going to differ because minerals that are breaking down into sizes that are directed by physical chemistry. Minerals break along certain planes, and so certain particle sizes are possible. whereas organisms are being driven by needs to have a certain cellular volume so that they can do their metabolic business and create energy. So what we have here in our sediment traps is that the purple is when we don't have a whole lot of cells in our system, and the green is during a bloom when we do have a lot of cells. So this wide swath of things shows that possibility space of all the ways that the minerals are breaking down, whereas the green is just showing that selected size range that is the optimal for the organisms in that system. So all of this, I realize now it's been a long time since we had a space picture, and I am from NASA. So this is a way to say that we're developing this as a way to look for life in something like a plume of Enceladus. Because with this conception of just thinking about particle size, particle chemistry, and fluid chemistry, you could use all those pieces of information that could be collected through a flyby to understand the probability that there's something alive in that data set. So that's where we're going with that main press. So yes, biosignature based on particle chemistry, size distribution, and fluid chemistry. But it's important to remember that particles are just really tiny surfaces. And this is also something you could apply to a surface. So this is some work I'm doing with my colleague Jeff Marlowe at Boston University, where we're looking at surfaces as a redistribution of materials that requires energy. We're looking at surfaces and mapping their mineralogical complexity and their elemental abundance. And we're doing that as a way of understanding that life preferentially will show up along gradients, because when you have a mineral interface, you can perform redox reactions and move electrons across that surface. And we take this information about the mineralogical complexity and the elemental abundances, and we can calculate the free energy of that surface. We can basically make what we call an energy landscape of that surface. And there's an old saying that Professor Jen McElady, that both me and Rachel know from grad school, would say, is that if there's delta G, something will live there. If there's life to be had and there's delta G, it's going to be there. And so we're looking at that idea and trying to use other tools as well as the mineral and elemental maps to see if that's true, to see if we can see that, and to see that across an age range of rocks. And so we start with very fresh rock. This is me in a lava tube in Hawaii, where we're mapping those surfaces. We're using XRF and LIBS to look at the mineralogy and elemental abundance of those surfaces. We're imaging them with Raman to see if we see any organic materials. We're collecting that rock, taking it back to the lab, and also looking for those same pieces of information with lab scale instrument versus field scale to try and build up those search patterns. And the reason we're using that suite of instruments is that's what's part of the Sherlock's instrument suite that's on the Perseverance rover. We're trying to build a search pattern to look for life with just those features that can be interrogated with high heritage instruments that we're currently flying and will certainly visit other planets and probably even Mars again. So that's the whole idea behind us using this sort of thing, using the tools at hand, kind of like the dust analyzer that will be around Enceladus. We're trying to build these search patterns for these tools that don't presuppose common heritage and common biochemistry of our own. So I know you guys heard in my bio that I'm an organic chemist. And so I'm sorry, there's no way we can go through the night without me talking about organic chemistry. I'm so sorry I have to do this to you, but I'm going to try to make it as approachable as possible, because a lot of these things are actually fairly intuitive. Just like we can look at this highly ordered structure with intrinsic complexity, showing this complexity of various scales in this living organism, we can see those same features of intrinsic complexity in the molecules it makes. In case you aren't a chemist, when you see diagrams like this, with these lines, that's just showing you things that are made of carbon, and at every node of this feature, that is a carbon molecule. This is a very familiar molecule to all of you. This is chlorophyll. This is the green color in plant. This is the light harvesting pigment in plants. This drives a huge amount of energy transfer on our planet. And it is without question, a fairly complex molecule. And when I say complex, what I mean is it is highly unlikely to arise on a landscape without the input of energy that we call life. That's what I mean when I say complex. Here are some other highly structured molecules with intrinsic complexity that we also associate with life. So when we think about looking for life on other planets, we say, well, we can just look for these complex molecules. We'll just look for, things that are big or have lots of doodads. Red in these conceptions is always oxygen, blue is nitrogen. So let's just look for those things, but that's a very unsatisfying way to start describing the chemical complexity of life, because we're making a whole lot of assumptions that the stuff that I make that's complex will be shared by organisms across the solar system. They'll be making the same complex things. Wouldn't it be better to come up with a metric that can mathematically describe chemical complexity when we say it? So that's what me and my colleagues at University of Glasgow in the Lee Cronin's lab have tried to do. And we started with the approach that without making assumptions about structure, is there a threshold above which molecules are unlikely to form without supporting biological machinery? And so we started looking at molecules like this and trying to move beyond, well, I'll know it when I see it. Yeah, that's complex. And so we started to think about a model where we could say, what is the simplest way to construct a molecule from its parts, including the simplifying feature of duplication? So that means if we took this molecule, naphthalene, and we just start putting two carbons together, and then a third, and then another, and then they come together in the ring, and the ring is a duplication event when those two come together. Will that number of steps that happen along that molecular assembly journey be a way to be an indicator of life? And the first thing that we did was torture a lot of metaphors to talk about this idea. We imagined shapes. We imagined Legos as a way to talk about steps. We imagined letters. And all of this is we were trying to represent this idea that assembly pathways are a sequence of joining operations where the basic building block is bonds. So if we think about banana as being this molecule we're trying to build, we got B, we got A, we got N. Our first step is to put B and A together. Our second step is to put N and A together. The third step is to put banana together. You add the banana, 4 steps, that's your number of joining operations. That is your molecular assembly number for banana. And we adopt this idea from graph theory. So now I've really offended everyone because I'm talking about organic chemistry and math. Everyone's going to leave now. But when we adopt this idea from graph theory, and we started to bodge those ideas around with algorithms that we could use for understanding graph theory and represent each of these different ways of understanding graph theory as chemical proxies. So edge weights, acids, reaction rates. And we generated 3 million trees of different properties to look at what, if you just give this algorithm all of the elements that you want, limitless abiotic precursors, all of the unconstrained chemistry it could ever want, what kind of molecules will it make? On all these random joining operations, will it start to make high molecular assembly molecules? And we found that those are rare in modeled space. Those things that have a whole lot of steps that we associate with you and me and chlorophyll, that's very rare in a random assembly of Legos, letters, and elements that are being joined into an organic molecule. Here is some of the organic molecules that we were trying to build and look at as steps. We've got asparagine, an amino acid with 7 steps, tryptophan with 12, penicillin and ATP, both things that we consider biosignatures, so they need organisms to make that. And we start to say, well, maybe just molecular weight is a proxy. Maybe we can use molecular weight as a proxy for MA. And so we then took a database, the Reaxis database, with 2.5 million unique organic compounds in there, and had it try to understand the molecular assembly number in an algorithm of all of the things in that database. And here's what we found. is that above a certain threshold, things actually diverge from molecular weight as just being a proxy for that molecular assembly number of steps. So this is, something that we can't actually just look at molecular weight and try to understand its probability to be related to life. So then the next thing we did was take that and just use the algorithm to predict that and try to find that threshold value for biogenicity by categorizing all of those molecules in that was table in that database, which was actually a little heavily weighted towards biological molecules. We started to recognize a threshold. It's about 15 or 16. So these molecules that are towards the bottom that have a lot of doodads on them and are large, those are the things that are very difficult to make without the presence of life, these larger things. And we started thinking, okay, great. We have a computer tell us things that are likely to be like life. How do we do that in space? If we haven't identified a molecule, how do we know if it's lifey or not? And so then we thought, well, we built a whole bunch of molecules through random walk. Let's try to break them down and see if they fall apart in the same number of joining steps as they were put together. And we use tandem mass spectrometry with fragmentation to do that. So we break apart molecules. We take this molecule right here. If you apply a certain amount of energy to it in an infinite, you can break it apart into these different fragments of different sizes. And that's what you're seeing here is the relative abundance of these different sizes of molecules from this parent molecule. And why this is something we wanted to try to do, why we wanted to try to artfully break molecules, is because tandem mass spectrometry is something you can do in space. We have ion trap mass spectrometers. Mass spectrometry is something we've done around nearly every planet in our solar system. And this kind of mass spectrometers that can do fragmentation like this, there's one that's already built for Mars, and there's one that's going to Titan. So it will be able to do this operation of breaking a molecule, counting this number of fragments, and being able to understand how that relates to MA by carefully controlling the energy. That's because biology generates higher complexity molecules, and higher complexity molecules produce more ions during fragmentation. We can see here these molecules ranging from not very lifey with an MA of 5 to really lifey with an MA of 27, and see the number of fragments generated there. Here is another version of that where we're taking a mixture of molecules, because unfortunately, when we go to other planets, it's usually not a pure product. We have to deal with mixtures. And so we started doing this, and what we would do is look at that biggest fragment, break it apart again, and see what the MA of that biggest fragment is. So what is the lifiest thing in this assemblage of molecules that we can use to tag the entire sample? And then we started doing that with stuff we found around the lab, like whiskey and beer and yeast. Sure. So this is a whole lot of dots and lines, but the thing to notice is that green, blue, and purple are all things that are or were alive. Things that are red and orange are things that have never been alive. And so when we use that fragmentation analysis, we see a whole bunch of stuff definitely rising above that threshold value of 15 or 16 that we think is that biogenicity threshold. We have some unfortunate things that should be a little bit more likely and they aren't, and that's because they're highly altered. We'll talk more about that. And I think for the benefit of everyone in the room, I just want to point out that the lifiest thing here is beer. Importantly, when we look at Murchison meteorite, this was our big test because meteorites have a lot of complicated chemistry. Is that going to be a red herring for life? It's actually a very unlikely rock. That's good. So it's working. So eagle-eyed viewers will have noticed a few things that didn't look like it quite worked. And one of those things is that there's a lot of molecules that have very low molecular assembly numbers that we associate with life. Things like amino acids, fatty acids, carboxylic acids. And so they're ambiguous. They show up in abiotic chemistry. They're made in stars. They're replete in meteorites. But they're also a big component of me and you. How are we going to get around understanding when we find an ambiguous molecule, if it's from life or if it's from abiotic chemistry? So what we've been working on in my lab is position-specific isotopes of those molecules. So that's looking within a molecule structure to see where there might be isotopes of an element that is heavier than the average weight of that molecule. And so we're looking in that weird low MA, high molecular weight region at molecules as well as these ambiguous things. And so here's just a representation of the kind of data that we're building as we have amino acids like alanine that come from Murchison meteorite. There are, just so you know, these things that we think are so special, amino acids, there's tons of them in meteorites, there's hundreds of them. And you and I as organisms are using a paltry 20 out of the hundreds of possible structures out there that we see in the solar system and meteorites. And then we took, we can look at organisms. There's 5 different organisms and see how the relative amounts of heavy isotopes of carbon are showing up either in these gray parts or the red part of this molecule. And so what you're seeing, as we would expect from a meteorite, is that a couple positions on that meteorite amino acid are highly enriched. Lots of the heavy isotope, whereas that never happens in things that come from life. And that's because these are abiotic reactions that are happening out in stars, and they have the benefit of all the time in the world on their hands. They don't need to be quick about these reactions when they're making them, and they have very different reaction pathways than the kind that biology is using to make amino acids, where those are enzymatic reactions that have to work at faster timescales, because you and I need all that alanine. We don't have solar system timescales to wait for it. I also wanted to point out to eagle-eyed viewers that yes, I am recognizing that there is a difference between complicated and complex chemistry, because there is a lot of complicated chemistry happening out in the interstellar regions with hundreds of molecules showing up. But interestingly, most of those that we see out in these regions are fairly small. But there's this recognition that tons of organic chemistry is happening out in space. It's not particular to you and I and all the other critters on Earth. And some of that can be very, very large. When I first started thinking about this, I've done a lot of work on PAHs, polycyclic aromatic hydrocarbons in meteorites, and those can be huge. But back to our idea from naphthalene that once you're joining things, that's no longer a unique joining step. That doesn't actually, if you're just putting rings together like a bunch of Legos, you're not actually adding any complexity to that structure. So just that's a caveat when thinking about space. Just thinking about the kind of organics that we see in meteorites, there are tons of them. On this graph, this is data from an FTICR. Every one of those lines that you see in this is a representation of a molecule showing up at that molecular weight. And these graphs are just showing us all the possible ways that those many, many molecules are putting together just a few elements. So this is a very complicated system with lots of molecules, but all of those molecules are pretty small with very low MA numbers. They're not rising to the probability threshold that we have for understanding the number of joining steps that organisms are doing in organic chemistry. I did also want to point out that there's over 10,000 discrete molecules, organic molecules inside meteoritic reservoirs. The last couple of things that I wanted to talk about to try to bring home this idea of selection mechanism and how organic molecules are showing their biogenic possibility versus their abiotic is when we look at isotope space. So along this axis is light to heavy carbon, along that axis is light to heavy hydrogen. And when we look at the meteoritic reservoirs, again, they're incorporating tons of that heavy isotope elements because they're just out there in space doing their thing. Whereas organisms tend to have the depleted amounts of both of those isotope systems. I'm going to end with this idea of amino acids, since they are so common in the abiotic systems as well as in organisms, and to bring home that idea of selection mechanism, that organisms have to make choices about how many of these molecules are going to make and maintain as part of the metabolism. So if we look in meteorites, we see tons of amino acids, 80, 100 amino acids. You and I only have 20. And amino acids are special molecules because they're chiral. And what that means is that they have handedness. They can be a mirror image, but are non-superimposable. So they have these geometric ways of putting themselves together that are very distinct. And out in space and in meteorites, both hands, both geometric forms of those molecules are showing up. Whereas in life, you and I, just the left-handed is showing up. And there's other kinds of structural diversity that have to do with how that side group is arranged on that amino acid molecule. And there's a huge bunch of ways that that can happen. And they're given dumb names like A through epsilon or alpha through epsilon. But in organisms, we only use the alpha structural form of amino acids. And again, we're preferentially using lighter isotopes versus space using the heavier isotopes. So all of this is just a way of saying that this represents that flux of matter and energy that was led by a selection mechanism and represents a redistribution of matter that we can call life. So I've tortured a lot of you with organic chemistry and math, and there probably weren't enough space pictures for all of you. So I want to kind of bring this idea, this journey that we've gone on across the solar system through time, to this idea of what are we really doing when we talk about life and talk about ways that we think we're going to detect it. And I always like to bring it back to the fundamental ideas that Andy Warhol was talking about in his work. His work wasn't just painting sloppy pictures of tomato soup cans and soup cans in general, or with laundry boxes and all sorts of things. It was a way of saying what is truly original and creative. What are the roots of creativity and originality, and how do we avoid just appropriating things as we say, oh, this is a good thing? And we can think about that same idea when we're trying to look for life. When we come up with a conception of what we think life will look like and a way to look for it in the solar system, are we truly understanding that system and the possibility space for life? Are we bothering to understand that abiotic context, that potential habitat? Or are we actually just appropriating patterns from modern Earth and calling that life detection? Notice how this life detection. And with this idea, this very esoteric idea about life detection. Thank you. Oh, and I will take questions. So the bad thing about that is you're assuming that proteins are what are building molecules everywhere in the solar system. So you're right. If we're looking for something on Earth, that's usually what we do. When we go out into the environment here on Earth, we're usually looking for those really common things. We don't go with analytical chemistry kits like the SAM rover or the Curiosity rover. We go and we sequence because we're making that assumption that's pretty valid that we're going to share all that same biochemistry. Yeah, that's a really interesting idea. And fundamentally, the idea of molecular assembly doesn't have to be carbon. We are trying to do this with mineral databases to understand, because, if you guys have read Bob Hazen's work about mineral evolution, there is a vast difference between the kind of minerals that show up on an abiotic surface versus a surface once there's life on it. So that is an interesting idea that we're trying to think about. Minerals are a little challenging when we think about silicate minerals, especially because even though they've got all the same number of electrons and stuff like that, are we seeing the same number of joining operations into really big things? And are they present on a landscape and in the map, as you can see? So we try to approach the inorganic space a little bit differently from just thinking about what abiotic mineral surfaces look like versus those abiotically, like in those lava tubes in Hawaii, versus those that have been affected by life. So the effect of life on a mineral surface is probably how we're approaching it right now. Yeah, so that's what we're actually looking for, is we're looking for the freshest surface. We're trying to understand, again, life as a comparison to the abiotic context. And that's really hard to do because, again, if there's delta G, a microbe's going to go there and live there. And so we have to usually take this kind of work down into the subsurface. We find lava tubes that we don't think are affected by sea spray and stuff coming in from the wind and, you know, other people, things like that. So we do try to find these places that are protected from the surface to try and look at those surfaces and understand that. We've done similar work in other cave systems, but you know what makes Hawaii unique is most of that rock is pretty fresh. If you look at rock in one of these protected parts, it's usually pretty mineralogically, I don't want to say boring, but homogeneous. And there's a certain amount of water rock reaction that can start to bring out interesting precipitates. You start to find gypsum veins and stuff like that. But at the point that you're starting to have water rock reactions and do interesting chemistry like that, you probably brought in life in a fracture water. And so it's a really tense threshold between actually being able to find out what abiotic rock looks like in its native state versus rock that is, you know, with life. So some of the ways that we try to do that, to get around that, is also treating meteorites the same way. Meteorites shouldn't have any life on them, and they do have a lot of mineralogical complexity, and they do have a lot of weird elemental abundances, but are they arranged in any way that represents an energy landscape, that they were gardened in any way by microbes to be good surfaces for actually extracting energy? And that's something that we don't quite see with those rocks, even though they're very heterogeneous and have lots of interesting chemistry going on. That's how we kind of approach it. Yeah, so that's actually really key to a lot of these reactions that we're seeing. As again, we're seeing a lot of the same cool chemical products coming out of space that you and I are making right now, but we're making them at a super fast rate, fast enough that we're not incorporating heavier isotopes into those products. So that's one way of trying to conceptualize the catalytic nature of life. And so that's something you wouldn't quite see. Another way to think about the catalytic nature of life is also just to think about the distribution of kinds of molecules that are made. Catalysis is making a product. Enzymes inside you that are catalyzing reactions right now are making a product. When we think about the crazy chemistry, I'm acting like there's still a slide behind me, that's happening in interstellar space and being picked up by meteorites, that's just Lego. That's just glomming things together into ever more randomly large, complicated structures. So that distribution space of molecules is also a recognition of the nature of catalysis. Okay, so that's kind of what we were trying to do with that benchmarking activity that we did for molecular assembly, where we took stuff around the lab, yeast, beer, whiskey, someone else's lab, and analyzed it to understand, you know, where it was in molecular assembly space. We're looking for the weaknesses. We're looking for things like those ambiguous molecules, which we clearly have to interrogate in a different way, not only for understanding life, but contamination potential, which is huge when you're not assuming what you're going to find. And the other way that we're looking at that is to try and understand all those other things that are fooling us. Like right now, and all the geologists in the room will appreciate this, is we don't have a really good way to use molecular assembly for altered fossil carbon stuff that comes from life. If you look at coal, coal looks dead. Coal is not dead. Coal is very much from life. And it would disappoint me to no end to send an instrument to Mars and it found coal and told us, sorry, back to Earth. I mean, it's actually something that's living. So we're really having to contend with those kind of things. And that's why we're digging more and more into the small molecules to look for those subtle differences. Good. So yeah, that's kind of what we're trying to do with those benchmarking activities. So this was actually an interesting idea that we have been working on, trying to use image recognition to see if we can look at surfaces with something like a confocal microscope, which can be put on a spacecraft and sent to another planet, and see if it would look at those sediment, as I said, geology accretes all of that pattern that life left behind. Would a microscope on another surface be able to pick that up? To be perfectly honest, it wasn't horribly successful. And what's disappointing about that is this idea of intelligence, that life has to recognize life, because there are probably paleontologists in the room, and you can all go to an outcrop and identify life like that much faster than an image recognition system. And how did you learn that? Probably your advisor yelling at you a lot and telling you when you were right or wrong. And that process of training algorithms isn't nearly as good as your advisor yelling at you. But we haven't figured out how to do that with technology yet.
I mean,
Yeah, so you answered your first question with your second, but what's different there is that energetic input that isn't, that you could sit down and do a calculation to think about what crystal growth is. It's much harder to do calculations that predict how biology reacts with matter to create energy for metabolism. So that's a much harder thing that brings in a bunch of extra components of energy. And so it's been a long time since I saw the Andromeda strain, but it's coming up a lot these days because of Mars sample return. And there's a lot of effort that we're putting into trying to recognize life that might not have the same biochemistry as us, that could be in those samples we bring back from Mars. A lot of thinking is that if it's so foreign and doesn't use the same energy systems or matter that we use, it's probably not dangerous to us. But we still want to know if it's there, because that's life detection right there. So we're using some of these agnostic methods and thinking about that first set of investigations we'll do with the Mars samples. So, fun fact, there's probably not a really good definition of life. Many groups have convened panels of wizened scholars to discuss what they think the definition of life is. And we haven't really arisen on anything much more satisfying than self-replicating chemistries that follow Darwinian evolution. That's even worse than metastable entities. Because I literally don't know what that is. So yeah, so there's operational definitions of life. Most biologists use operational definitions of life, but there's not really a definition of life per se. Lots of people get mad about this and want NASA to decide, and that's not our business. And so, you know, I don't think it actually hampers this that much though, because if we think about the business of physics, especially big physics, when people are looking for crazy particles that they don't quite even know are there, they don't actually have definitions for a lot of those things. So in science, we actually do look for things that we don't quite know what they are all the time, and we haven't gotten too upset about having to define them. Yeah, so right now, by agreement within the community, life detection has become a really important goal for the agency, for NASA. And the places that we're really focusing on right now for future investigations are Enceladus, so the ocean world that I showed you. There's a lot of great reasons to think there might be life there. It's got a lot of the same solvents, it's got energy sources. It's pretty nutrient poor, it's energy poor, you know, but it appears to be a place where you could harness energy and perform reactions in a way that sustains life. The other place is there's a real move to start reinvestigating Mars as a place that might have extant life there right now. Yeah, we're actually keeping a really broad search pattern. So if you think about, you know, the idea of particle chemistry, particle size, fluid chemistry, that could be anything. You know, we're just looking for distributions of elements that, you know, aren't equilibrium physics like we would like to see it in a fluid. So yeah, so we're trying to keep the search patterns as wide as possible. And that's something that I think a lot of scientists in this field and NASA are really invested in right now, is trying to keep that search pattern as broad as possible. Yeah, so. Oh yeah, so there's Martian meteorite. lovingly referred to as ALH 84001.
4001.
All the grad students who came to lunch with me, I told them in depth why it was named that, and I think they were bored out of their minds. But this meteorite had weird structures in it that looked like something that could be a fossilized microorganism. And scientists that were working on it were so convinced that there was an announcement at the White House that we found a Martian meteorite that looked like it could have life on it. And of course, that means that like everyone in science was like, I want a piece of that and look at that. And so the evidence didn't actually stand up. There was a lot of other things that happened after that to show that the carbon within that was more graphitic. But this is an interesting thing to talk about, not as some sort of failure as astrobiology, but as science working in the way it should. Somebody made a supposition and then, everybody went and tested it using their particular tools, and it's gone back and forth in the literature for a long time. And so it's really, science working as it should, other people checking each other's work and bringing new information to the table. Now, we haven't done anything like the test that I've talked about with something like the ALH 84001 meteorite, although someone in the consortium that I'm the deputy PI of has recently published work on this meteorite. And I think, we will, as these techniques mature, want to do something like that with that meteorite. This is not something we've done yet. Yeah, so this is an interesting idea, and I'll talk about the defense of carbon as a basis for an organism to address this, because it sounds like this is a lot of interest, like what other element systems are there? And part of the reason why we think that organic chemistry would be a really good place to start is because when we look out in space and see the kind of chemistry that's happening and imagine all of those materials that would be on early planetary surfaces, we look at what might be soluble in fluids on early planetary surfaces. And the most likely fluid, sorry, Titan, is stuff like water. Water's all over in the solar system. We keep on finding it on Mars, big deal, it's everywhere. So when we look at these early planetary materials and try to imagine what life would come from, we often try to think about what would be soluble in water. And so that's where the importance of looking at soluble organics and meteorites comes in. So that's kind of the defense of that idea, is it's something that would be there in probably big amounts, soluble. You need to have something soluble if you're going to start doing interesting chemistry. And if we think of prebiotic chemistry and what that really means, is it's just nature doing a whole lot of experiments with what it has on hand, and until maybe something interesting called life arises from that reaction mixture. Now, there is lots of other stuff that's soluble, and you could start to think about that, but does it have the same complexity of chemical forms showing up that give you lots of options, lots of different reagents to tinker with in your lab? Not so much, not when compared to organic chemistry. So that's kind of the defense of why we think of organic chemistry. And when I say that, I mean reduced carbon as being a really likely thing for life to come from, just because there's lots of different materials you can do experiments with. That's not to say it's the only way. It's just something where there's a lot of options.
And I have had Heather come give a talk in my class, and she stayed for 45 minutes of questions. So I know this could continue all evening, but in the interest of everyone getting home safely at some point, I just want to once again thank you, Heather, for sharing your research, your passion for science and space with us, and coming to visit us when it is literally the coldest day we've had this entire time. So thank you very much.